All right, hello, welcome back. Before I get into this today, I just wanted to sort of apologize for the background noises that you're probably picking up in these videos. I tend to record these pretty early in the morning, so I'm usually just sitting in my living room drinking some coffee. A lot of times I've got the window open, so you're going to hear birds chirping and cars driving by, and my dog and cats are probably chasing each other around. So anyway, if you're hearing weird stuff going on in the background, that's probably it. Um, so today we're getting into chapter three, and really chapters three and four are kind of tied together. So in this section, we're going to be talking about macromolecules, and I'll go into what those are, but these are really these just biologically critically important molecules that you're going to find making up all living cells. So these are going to be proteins, carbohydrates, lipids, nucleic acids. So we're just talking basic structure and function here. And in chapter three, there's a lot going on because it's also got the just background introductory information on macromolecules. Um, since there's so much going on, I'm going to have this divided into probably at least three parts for chapter three. All right. So, like I said, we'll, excuse me, start with a little bit of background. Um, you know, what are these macromolecules? Why are they so important for living things? And then we will start talking about basic structure and function of proteins and structure and function of carbohydrates. All right, so again, we have these four main categories: proteins, carbohydrates, lipids, and nucleic acids. And most of these, with the exception of lipids, are what are considered polymers. And so polymers are large molecules made of repeating subunits called monomers. Excuse me, got a tickle in my throat. So you will take these monomers, think of these as the building blocks, and link these together to form these long chains of polymers. Now, I mentioned lipids are not polymers, but the other three categories of molecules here are. So proteins are made out of monomers called amino acids, and there's 20 different amino acids that are used to make proteins. Carbohydrates are built out of monomers called monosaccharides or simple sugars. We link those to form polysaccharides. That would be the polymer form of carbohydrate. And nucleic acids are built out of monomers called nucleotides. There's four different nucleotides. So, if we look at the distribution of these four classes of molecules in living cells or in living organisms, we see this phenomenon called biochemical unity, which is that we find basically the same proportions of these things in all living cells. So if you were to take a cell from pretty much any organism and just do a basic biochemical breakdown, you would find, approximately a quarter of that cell is made up of those macromolecules, those four types we were just talking about. Um, of course, the vast majority of the cell is going to be water, and then you have a little bit that's going to be other ions and other small molecules in there. And if we focus in just on the slice that's macromolecules, you're almost always going to find this particular distribution. So a little bit more than half of the macromolecules are proteins, about a quarter of those are nucleic acids, and then you've got smaller slices of lipids and carbohydrates. So it's kind of crazy you find the same distribution in all living cells. And really, what that would argue is that because there's certain things that all cells have to be able to do, certain functions that are similar across all living cells. Um, to carry out those functions, we need these macromolecules in these particular proportions to be able to accomplish those jobs, those functions. So biochemical unity. All right, so coming back to this idea of polymers, as I mentioned, you build polymers by linking monomers together. And you do this by forming covalent bonds. And you remember in the last chapter we talked about the fact covalent bonds are just two atoms sharing electrons with each other. Now, anytime we join two monomers with a covalent bond, this is considered a condensation, or sometimes you'll see it called a dehydration reaction, um, because we basically remove water. And what I mean by that is, so here's our two monomers, just kind of generic cartoon chemical structure. But what we will do is we will remove this OH hydroxyl group from one of the monomers, remove a hydrogen from the other monomer. Those come together to form an H2O, right, water. That's quote unquote removed. And then we are left sharing electrons between the remainder of those two monomers. And so that's the formation of our covalent bond there. And these polymers are always going to be big molecules, so we're going to link lots of these monomers together. So we bring two together, dehydration reaction, remove the water, form that covalent bond, bring in a third monomer, another dehydration reaction, remove the water, form another covalent bond, and so on. So we could just build this long chain of these monomers linked together, and that is our polymer. And if we wanted to start with a big polymer and break it down, we do that with what are called hydrolysis reactions. And you see this term a lot in biology and biochemistry, and that literally just comes down to breaking water. Lysis is break. And so we're breaking these covalent bonds in the presence of a water molecule. So it kind of makes sense. If we removed the water to form the bond, we basically have to put the water back in to undo that, to break the bond. So we can see here's our polymer, right? We bring in one water, we can break one bond, bring in another water, break another bond. And so we're going to see examples of this throughout the coming chapters, like building these big macromolecules up, and then in some cases breaking them down, maybe to release some of the energy that's being stored in those bonds, but we'll get into that later. Okay, so hopefully you feel comfortable with that basic idea of a polymer. And I've been using this term macromolecules, large molecules. What does that really mean? Well, most biochemists are going to define macromolecules as those that have molecular weights of greater than 1,000 kilodaltons. And so you might get the impression, just based off what we've talked about so far, that, well, these polymers look like they're going to be just really boring, uniform molecules without much diversity, right? We're just linking these repetitive monomers together. And while that can be true, that obviously was not going to cut it in terms of biology, right? Because living organisms are very diverse. Even different cells within one organism may need to be very diverse, doing lots of different things. And most biology is being powered by these macromolecules. So how do we get that diversity? And I want to talk about a few different ways we can do that. But one is by adding what are called functional groups. And so you may have seen this term before, but functional groups are just these chemical groups that could be added or removed from molecules that are going to bestow certain chemical properties upon the molecules that carry them. So you could have functional groups that are polar or nonpolar or acidic or basic, and by adding those to one of these polymers, we make that polymer polar, nonpolar, acidic, basic. So yeah, so you might see one macromolecule, one polymer that will have a lot of these different functional groups. And so I'm going to go through here just a short list of some of the most common functional groups. And I'd like you to be able to, just know the names of these and recognize them by sight. And then also just be familiar with the basic properties that these functional groups give to the molecules that carry them. So for example, the first functional group here is the hydroxyl group, that OH group that I just mentioned a minute ago. And hydroxyl groups typically characterize alcohols. So anytime you see a molecule like here, you've got ethanol containing one or more hydroxyl groups, there's a good chance that that molecule is going to be considered an alcohol. The second functional group is what's called the carbonyl group, which is carbon double bonded to an oxygen, and there may be a hydrogen attached as well to that carbon, depending on whether or not the carbonyl group is at the end of a molecule or in the middle of a molecule. But remember, those double lines there are representing a double covalent bond, so the carbon sharing two pairs of electrons with that oxygen. And carbonyl groups are really reactive, so we tend to see carbonyl groups in some of these important energy reactions that are going on in cells. And we're going to see these pop up a lot when we talk about cellular respiration in an upcoming chapter. And if you're not familiar with that term, right, that's basically the process that goes on in your cells by which sugars are being broken down and the energy released and used to build ATP molecules. So when you see carbonyl, think carbon double bonded to oxygen, think energy, energy rich molecules involved in some of these energy releasing reactions. So we got hydroxyl group, carbonyl group. Third one on the list is the carboxyl group, and if you're going to get any two of these confused, it's probably going to be carbonyl and carboxyl because the names are similar, and in fact the groups themselves have some similarities, right? You'll see in the carboxyl group, again, you have a carbon double bonded to an oxygen, but in the carboxyl group, you're also always going to have a hydroxyl group coming off that carbon as well. And the key property I want you to know for the carboxyl group is that it is an acidic group. So any macromolecule that carries a carboxyl group is going to tend to be acidic, and that is because this group basically releases those H plus ions when put into an aqueous solution. And remember, the more H plus ions that are floating around, the more acidic that solution is going to be. Then we have the amino group, nitrogen bonded to a couple of hydrogens here, and this is a basic group. So carboxyl was acidic, amino is basic, and again, it is basic because it will accept H plus ions that are present in the solution, and this NH2 will basically get converted into NH3, so basically capable of forming one more covalent bond here and soaking up an extra hydrogen. So taking that out of the solution is going to make it more basic. Excuse me. And last two I wanted to mention here are the phosphate group. You see this a ton in biology, and we're going to bump into it all the time this semester in various chapters. But it's easy to pick out of a lineup. You've got the phosphorus double bonded to an oxygen and then bonded to three other oxygens. It's also an acidic group. And you also really commonly see this associated with energy-rich molecules. So for example, ATP, which is that classic energy molecule in cells, is adenosine triphosphate. It's got three phosphate groups attached to it. And the last one is what's called the sulfhydryl group, this SH. And honestly, this one's probably the one we're going to see the least, but it is going to pop up here later in this chapter, so I wanted to mention it. But this is really important for protein folding. If you have a large polypeptide, right, a protein polymer, these functional groups help that polypeptide fold up into the appropriate 3D shape, which we'll talk about here in a few minutes. Okay, so we can add diversity to these polymers by adding one or more of these different functional groups. Another way we can get diversity is through what's called isomers. And isomers are defined as molecules that have the same chemical formula, but the atoms are arranged differently. So for example, we've got examples at the bottom of the slide here of two different what would be called considered structural isomers. And so we got butane and isobutane. And when I say they got the same chemical formula, these would both be what? C4H10, if I'm counting that right. So both have the same number of carbons, same number of hydrogens, but as we can see, those atoms are arranged in a different pattern, so they're structurally distinct, and that will give them different chemical properties. Another common type of isomers are what are called cis-trans isomers. So these are isomers where you have different orientation around a carbon-to-carbon double bond that's typically somewhere in the middle of that molecule. So if you have whatever's happening on opposite sides of that double bond kind of oriented in the same direction, those would be considered cis-oriented. Whereas if whatever's going on the opposite sides of the double bond are pointing in opposite directions, those would be trans. So again, we can see these are isomers because they have the same chemical formula, but things are just oriented differently around this central carbon-to-carbon double bond. So we call these cis-trans isomers. And the last kind of isomer I wanted to mention is what are called optical isomers. And these are really mirror image molecules, so think left, right hand. And these are pretty common. So typically in these optical isomers, you'll have some central carbon, and then whatever groups are coming off of that central carbon are just arranged asymmetrically, so in a mirror image fashion to each other. And something like this can seem really subtle, right? If you at first glance you're like, oh yeah, those two molecules look about the same. They should have basically identical chemical properties. But it turns out even those little differences can actually cause big differences in the function of those molecules. So for example, there's all kinds of drugs out there, like ibuprofen is a good example of this, where when you produce ibuprofen, you get two different isomers being made. I believe they're optical isomers, one of which has this great biological activity, works as a fever reducer and pain reliever, anti-inflammatory. The other optical isomer has no biological activity at all, so it won't do anything for you. So you really got to make sure you're getting the correct isomer purified out of that mixture. All right, so we've got this base now, about polymers being formed from monomers. You've got some ideas on how you can add diversity to these polymers. So what we're going to do in the next section here is start talking about the different groups of macromolecules individually. And I want to start with proteins, which are arguably maybe the most important, or at least the most, I would say, interesting of these groups, because proteins are really carrying out all of the important jobs in living cells and living organisms. Proteins can be enzymes, right, speeding up chemical reactions. There are structural proteins, defensive proteins, transport proteins, really you name it. If there's something that's happening inside of one of your cells, inside your body, it's probably protein that's making that happen. And as I alluded to earlier, proteins are polymers. They're made up of 20 different amino acid monomers, and we link these amino acids together to form what's called the polypeptide chain. So that would really be the polymers, that polypeptide chain. And this is just a long, straight, unbranched chain of amino acids. But to really turn that polypeptide into a functional protein, we've got to fold that chain up into some specific three-dimensional shape. And what that shape is going to be different for every single protein. So I'm not going to force you to memorize the names or structures of of all the amino acids. If you're a science major, you may have to do that somewhere down the road. But I do want you to know this basic generic structure that amino acids have. So all amino acids share this structure, which is they have a central carbon called the alpha carbon. They always have an amino group and a carboxyl group. So carboxyl group is giving them the acid part of their name. And then what makes each of these 20 different amino acids different is the R group here. So this R group could be something as simple as just another hydrogen, or it could be some big complex chemical group. I'll show you a few examples here in just a second. But each of the 20 different amino acids that we use to build proteins has a different R group. So yeah, here's some of these examples. And in the next couple of slides, you're just seeing some of these amino acids just grouped based off of the chemical properties of those R groups. So for example, here are 3 amino acids: arginine, histidine, lysine. You don't have to memorize these structures. Where the R groups all have a positive charge somewhere in them, right? Positive, positive, positive. So those would be positively charged amino acids. And we've got a couple of negatively charged R groups over here, and so on. But you can see, right, some of these R groups are really simple. Like I said, just a hydrogen. Some of them have these big kind of complex ring structures, so be pretty diverse. So 20 different amino acids, 20 different unique amino acid chemical properties. All right, so what does it look like if we link 2 amino acids together? So remember, each of these would be considered a monomer. We're going to have a dehydration reaction, condensation reaction, as I mentioned before. So we're going to remove a water. You can see where the water's coming from, labeled in blue here. Right? We remove that H2O, and then the carbon on one amino acid is going to share a pair of electrons with nitrogen on the other amino acid, and we're going to get this covalent bond formed. And because proteins are so important, we give a special name to this covalent bond and call it a peptide bond or a peptide linkage, but it's just a covalent bond, just shared electrons. And I also wanted to mention here, the 2 different ends of a polypeptide, right? So here we have a very short polypeptide, just 2 amino acids, but this would be the same if you had 1,000 amino acids linked together. The 2 ends are going to be distinguishable from each other just based off the chemical groups that are kind of hanging off each end, right? So at one end you have that amino group, so it's called the amino or the N terminus of the protein, and then at the other end you have the carboxyl group, so C terminus. You might be thinking, wait, that's not a carboxyl group, it doesn't have the OH. Well, we lost the H here in that aqueous environment, but it is still considered a carboxyl group there. All right, I'm going to stop there and I will pick this up in part 2.